with my internet connection this afternoon, no? So let me share my PowerPoint. Okay, so can you all see my PowerPoint? Kindly confirm if you can see it. I think,
Yes, sir. We can see
sige,
that.
makita ra sa tanan. No? Uh, at the end of our class this afternoon, I will be checking your attendance ha. O niya, just say present if your name is called. So, masin makatugang uban diha. Okay, so for this afternoon, we shall be starting the lecture proper for our microbiology course, no? So we will be having this particular contents, no? We shall be introducing the discipline of microbiology, and then the first major content of discussion for this afternoon would be the members of the microbial world, and then we will go into evolution and diversification of microorganisms, and then the evolution and advancements in microbiology. Okay. Now, let us define the discipline of microbiology. No, so microbiology is a part of biological science, no, wherein we are dealing with the study of microbes. And when we say microbes, these are very tiny microorganisms or species, no, that are too tiny for us to see with the use of our naked eye. No? So eventually, we would need a certain device for us to examine the morphology, the structure, as well as the behavior of these organisms for us to be able to understand their biology. Now, how do we consider a particular organism as a microorganism, no, or as a microbe? So the term microbe is very broad, no? So it would encompass several groups of organisms that are present in our natural environment. So for us to really pinpoint that this organism is a microorganism, we have a particular criteria based on their size. No? So one major category for us to really see that these organisms is a microorganism is their size. No? From the name itself, micro, that means very tiny. No? So if, for example, the organism is unicellular, then it must be 1 millimeter or less in terms of diameter. So if they are fit on that particular criteria, then we may consider that unicellular as a microorganism. And if they are multicellular, no, meaning these are actually aggregates of different independent cells that are working together to form a singular functional unit. No, and uh, when you have a multicellular microorganism, usually or majority of the times, they are eventually colonial in nature, meaning they are independent cells that are living together in a community. No? So they lack this what you call differentiated tissues. For instance, in humans, di ba, we have epithelial tissues, no, muscular tissues. So they don't have those differentiated tissues. When you say differentiated tissues, these are a group of cells that are actually having different specializations, but then they are functioning for a single purpose. No, so they don't have those differentiated tissues. Now, in terms of classification, your microorganisms, no, the classification of microorganisms are kind of problematic in the past, no, most especially during those time of Carolus Linnaeus and before the time of Carolus Linnaeus, because their way of classifying things is based on morphology, no? And since you are now basing the classification on morphology, you're actually expected that the life form or the species or the individual itself can be seen by your naked eye. So that's the problem there, because the microscopic world is hidden behind plain sight. So dili na to siya makita uh, on a daily basis or regularly or commonly with our naked eye. So therefore, during their time, the classification only limits to the groups of being a plant or an animal. So that's the difficulty and challenge during those particular times. And another challenge there is that your microorganisms are very much diverse in terms of their characters. And these diversity in characters often lead to them being not classified as plant nor an animal, because most of these or majority of these microbes do have characters that are a mix of plant-like and animal-like characteristics. So uh, for example, one particular problem is that microbes cannot be classified in either the plant or animals, because both groups are multicellular. So they don't have representations of organisms that are unicellular, and most of our microbes are made up of only a single cell. Another challenge would be microbes may have a combination of features that can be found in both groups. As I said, for example, some microorganisms are quite motile, just like your animals, but they do have cell walls, just like your plants. So they really have this mix of characters coming from plants and animals. Some might have also, for example, um, in terms of their um, getting of nourishment, some might be, for example, you may call as mexotroph, mix troph, meaning there is a combination of different nutrient getting or acquisition mechanism. For instance, an organism which is both photosynthetic and non-photosynthetic, meaning phagocytotic, is a mexotroph. When you go to euglena, for example, euglena, they do have this green pigment chlorophyll, so they can photosynthesize. However, in the absence of sunlight, they can also devour other unicellular organisms. So in that way, they can manufacture their own food, but at the same time, if sunlight is not present, then they can also eat other organisms. So it's it's a mix of different characters coming from trees and your animals. So it's really quite challenging for your microbes to be considered as a plant
Or
or
an
an animal.
animal.
So nothing in between. And when we have a background when it comes to microorganisms, your microorganisms are the organisms that are in between your plants and animals. So the breakthrough in terms of cellular architecture, because of the use of, for example, we now have scanning electron microscope, very powerful microscopes. We're now able to advance our understanding in terms of microbial taxonomy, because we are now able to see the intricacy and complexity in the structures of a particular cell. So we are not only able to classify your organisms as plants or animals, we are now able to differentiate the very cells that are comprising these life forms. So when we now go to cells, you have two types of cells, diba? You have the prokaryotic cell, and then you also have the eukaryotic cell. And we can differentiate them based on the presence of nucleus. That's the simplest definition of being a prokaryotic and a eukaryotic cell, because when you have prokaryotic cell, that means that you do not have the nucleus. And if you are eukaryotic, then you have the presence of the membrane-bound nucleus. And because of the advent of these particular microscopes, and we are now able to see these intricate details. And that also leads to the creation of the Five Kingdoms scheme by Robert Whittaker, because we are now able to differentiate the cells. Then that would also mean that these organisms with different cells are actually grouped together in no, not grouped together, but they are grouped separately. So in the Five Kingdoms scheme of Robert Whittaker, you now have what? The kingdom Monera, for your prokaryotic representatives. you have kingdom Protista, kingdom Fungi, Animalia, and Plantae for your eukaryotic representatives. No? So eventually, we now included, no, there is now an inclusion of our microorganisms in these five kingdom scheme. And out of five, Monera, Protista, and Fungi, they represent the microbial world. No? But currently, um, the what you call this one, the classification Monera is not anymore uh, accepted by many scientists because we already know that the cellular structure of bacteria and archaea are quite different from one another. So therefore, from that kingdom Monera, it was divided now into two kingdoms. So you now have the kingdom bacteria, eubacteria rather, and the kingdom archaebacteria. So currently, what we are actually using right now is the six kingdom scheme of Carl Woos. So we have from Monera, we have kingdom eubacteria and kingdom archaebacteria. And from your eukarya group, you still have the kingdom protista, kingdom fungi, kingdom plantae, and kingdom animalia. Now, but we started with uh, only two kingdoms, di ba? So, sa time ni Carlos Linnaeus, because he was the one who proposed to us the taxonomic hierarchy, you only have the Plantae and Animalia. And then here comes Haeckel. I don't know if you know him. Ernst Haeckel was actually the first one who coined the term ecology, no, the study of interaction of your living organisms with its natural environment. So Ernst Haeckel now introduced to us the kingdom Protista. And from there, here comes Chaton, no, wherein he classified the life forms not only because of the behavioral and um, acquisition of nutrients, no, photosynthetic or not, but he eventually classified life forms according to the types of cells. No? So you have now your um, prokaryota, no, and you also have eukaryota. But these are actually another taxon. Or when you say taxon, those are groups that is not a conventional group in the Linnaean hierarchy because um, in Carlos Linnaeus' taxonomic hierarchy, he was not using the term empire, no, and then here comes Copeland now. Also introduced to us Monera, Protista, Plantae, and Animalia. And then Whittaker now introduced to us the kingdom fungi. No? And then several years later, we do also have what you call the three domain systems, no? Which is now comprised of your domain bacteria, your domain archaea, and domain eukarya. Okay, but in terms of our six kingdom scheme, no, one, two, three, four out of six are representatives of the microbial world. So, what are these members now sa ato ang microbial community? So we have here, no, a cellular based life forms and a cellular or non-cellular biological entities. Okay, so under your cellular life forms, meaning they are made up of cells, no? You now have your kingdom archaea, no? You have your kingdom eubacteria, you have your kingdom protista, and your kingdom fungi. So for your kingdom bacteria, these are now your prokaryotic. These are unicellular organisms with peptidoglycan molecule present in their cell wall. No? So when you say peptidoglycan, this is actually a very complex carbohydrate, no, made up of two different sugars. So you have NAG and um, what's the other one? The NAM. No? So NAG, that means N-acetyl glucosamine. And when you have NAM, that means N-acetyl muramic acid. So these two particular sugars are present in the cell walls of bacteria, which now comprise the molecule peptidoglycan. When you go to archaea, so your archaea is um, prokaryotic again, and just like your bacteria, it's unicellular organism, but they do not have peptidoglycan in their cell wall. What they do have is what you call a pseudopeptidoglycan. So your pseudopeptidoglycan is just the same with your peptidoglycan. However, the NAM or the N-acetyluramic acid is now replaced with NAT, no NAT, your N-acetyl tolus aminuromic acid. No? Lisod kay panglitok kun nga mga compounds, no? But you can eventually simplify your pseudopeptidoglycan as NAG plus NAT. And your peptidoglycan is NAG and NAM. No? When you go to what else? Kingdom Protista. So for your Kingdom Protista, these are now eukaryotic and unicellular. But some of them are actually colonial in nature. No? So when you go to, for instance, green algae, no, your kelp, what else? Your brown algae, no? These are eventually multicellular, but most of them are colonial in nature, no? Aside from your protista, you also have kingdom fungi. So your kingdom fungi is the very kingdom that represents both unicellular and multicellular microorganisms. No? So unicellular organism in your fungi is in the form of yeast. No, Saccharomyces cerevisiae, the one that you are using in kanang bread making, no, paghimo og pan, ang butang nato mga baking, no. And then for the multicellular representative for kingdom fungi, you now have the molds and the mushrooms. No? So eventually, mushrooms are not considered microorganisms, but molds they are considered as microorganisms. No, although sometimes if your molds are kinda really mature, no, pila na ka days nga nitubo sa inyong hangpan, for example, we can eventually see the colony of your molds. But if you try to look into the singular units of the mold colony, no, the hypha, for example, these are microscopic in nature. No, thus, they are also considered as microorganisms. Now, when we go to the acellular biological entities that are members of the microbial world, you have these four. No, you have viruses, you have viroids, you have satellites, and then you also have prions. When you say viruses, I know that you are very much familiar with viruses. No, so when you say viruses, these are acellular biological entities, meaning they are not made up of cell, and what they are made up is only a protein covering, which you call as capsid, and you also have genetic material that can either be an RNA or a DNA. No? So that's what comprises our virus. But some virus, they do have what we call an envelope layer, no, which protects, or an outer layer outside the capsid. And these envelope, they do have now what you call spike proteins. Now, for example, if you try to imagine your coronaviruses, no, these are the very viruses with the envelope layer. No, so as instead or aside from having the protective protein covering capsid, they also have the outermost membrane in the form of an envelope. When you go to viroids, no, your viroids are comprised only of RNA, and they are found only in plants, no? Not found in plants, but they parasitizes plant individuals or plant species. Okay, when you go to satellites, your satellites are often made up of RNA, no? Often, so not all the times they're RNA, they could also be DNA. But the main difference of your satellites from your viroids and viruses is that they need another virus, which you call a helper virus, that will help them to enter and invade a host cell and for them to replicate. So they cannot eventually replicate on their own. Your satellites need these helper viruses for them to be able to successfully replicate themselves. No? And then you have prions. Your prions, I don't know if nakadungog na mo sa term na prions, no? But these are misfolded proteins that can be induced that can induce misfolding in normal proteins. So, mo sila ang mga proteins nga nangahiwi ang structure. And take note, if you have a misfolded protein. Therefore, these are non-functional proteins. No? So, dili sila mo function. But in this case, since these are infectious type of misfolded proteins, if they attach themselves to a normal protein, no, a protein with normal structuring, then they could induce misfolding as well. So makatakod sila. And if that happens, this might lead to cellular damages, most especially in the central nervous system, as well as certain types of diseases, no? Neuro diseases, no? Most especially kanang maigo jud niya ang brain and the spinal cord. Example of diseases that are caused by prions are what you call uh, mad cow disease, no. I think nakadungog na mo ana nga sakit sa mga baka, no. Your mad cow disease, or what you call bovine spongiform encephalopathy, no. So ang ilahang brain ana kay mura siya sponge kanang daghag buslot. And then another disease that are kanang caused by prions in humans na kay gitawag nga kuru, no. I don't know if you're familiar with that, but kuru is prevalent in Papua New Guinea, no, due to ritualistic cannibalism. So in Papua New Guinea, historically, tribes there would eventually eat the brain of their deceased or dead relatives para daw ang soul sa ilahang dead relatives kay ma-retain silang family, and all the memories kay ma-handed down to the next generation. So that's the purpose of eating the brain. But then, not knowing there are certain misfolded proteins in the brain nga inikaon, maapil na noon ang ilahang normal proteins nga mo misfold, no. So kuru could be, the symptoms of kuru would be kanang catatonic state, no, kanang mukalit lag kirig, and zombie-like symptoms, no. So kanang murag wala na sa pangisip, and that's kinda dangerous, no. But currently, I think there is now a decrease in the reports of kuru in Papua New Guinea, but still there are certain reports of kuru disease because some prions might become dormant for a very very long time, no, up to 50 years. So dili ka kaingon nga dili na siya mo emerge o balik ba. And there are also some prions or misfolded proteins that are spontaneous, no, meaning dili sila inherited, dili sad sila nakuha sa gikaon, but they spontaneously being created or made inside your body. So there is really a certain degree of mutation, no, in the proteins. So these are just, no, a quick survey of the members of our microbial world. So naadjug kay unicellular or multicellular microorganisms. And we do categorize our microorganisms based on the size. Now, let's go to the diversification, the origin, no, of our life form as well as the microbes, no. So akasa man jud sila ni originate o ni start og unsa ni kanang being present in terms of geologic time frame of our planet Earth, no. So when you recall your systematics class, di ba, we have the geologic time frame, no, and the time frame of our planet Earth is basically subdivided into different time periods. Excuse me. You have aeons, you have eras, you have periods and epochs, no. So when you say aeons, these are um, billions or hundreds of billions of years. And then when you go to eras, these are tens of millions or hundreds of millions of years. And when you go to periods, these are millions or tens of millions of years. And your epoch is several millions of years, no? So from aeon down to epoch, kanang gamay ang imuhang kanang time frame. But when it comes to your aeon, we have approximately how many aeons? We have 4, no? So you have Hadean aeon, you have Archean aeon, you have Proterozoic aeon, and then you have the Phanerozoic aeon. Your Hadean, your Archean, and your Proterozoic aeons, these are eventually collectively known as your Precambrian period. No? So sa Precambrian, naadiha ang inyong Hadean, Archean, and Proterozoic. Now, approximately, it was kanang estimated that our planet Earth was formed 4.6 billion years ago. So from 4.6 billion years ago up to around 3.6 billion years ago, no, that's actually the time of the Hadean era. Hadean is named after the god of the underworld, no, Hades. And during this time, ang planet Earth nato kay dili pa siya unsa ni susceptible, no, not susceptible, but it's not sustaining for life, no, and maintaining for life. So eventually, it's very toxic, no, wala pa oxygen. It's really an adverse environment for life forms as we know it today, no. And after which, after the Hadean Aeon, then we have the Archean Aeon, where it was believed that the first unicellular organism has evolved. No? That's about 3.4 to 3.6 billion years ago. And from Archean Aeon, no, the first unicellular, at the last and the onset on the next Aeon, which is Proterozoic, it was believed now that your eukaryotic type of life forms had evolved. No? So, nag-una daw ang inyuhang prokaryotic. This was followed by your eukaryotic. And on the onset of the Phanerozoic Aeon, there is now diversification of complex life forms. Then you also have the evolution of multicellular organisms, no? And you have what you call adaptive radiation, no? Because you now have empty na mga habitats that are just waiting to be colonized by new species or new forms of life, no? So that's adaptive radiation. And under your Phanerozoic, diba, you have three particular eras. You have the Paleozoic era, you have the Mesozoic, and then you have the Cenozoic era. So when you say zoic, that means life, no? So when you say Paleozoic, paleo meaning ancient life, Meso meaning middle life, and Cenozoic meaning recent or new life, no? So all of these era under Phanerozoic are eventually dominated by complex forms of life form, no? So Paleozoic, you have ample amount and diversification of um, invertebrata, no, from your insects to your arthropods, mollusks, sponges, and then when you go to Mesozoic era, this is now dominated by your reptiles, no. So in your air, you have pterosaurs. In your land, you have dinosaurs, and in your oceans, you have mosasaurs. And then you now have your Cenozoic era, after which you now have your animal, no, not animal, but mammal-like reptiles, no, which dominates your Cenozoic era. And then 10,000 years ago, there is now the evolution of man. So eventually, from the simplest form of life, from unicellular prokaryotic organism, it now evolves and paved way to the evolution of man and the most complex of all life forms. So eventually, if we try to trace back all of these uh, species that we have, extant species, and even the extinct species, then we shall be going back to a microbial life form. No? So, kitang tanan, anak dito sa bacteria. And then, during that time, no, diba, during the Hadean Aeon, the environment is quite adverse and it's not sustaining and maintaining any forms of life. So there might be one particular point in time as to where it was believed, no, prebiotic time, meaning wala pa'y cell, wala pa'y life form, it was believed that a certain molecule has evolved its function and capacity to catalyze certain chemical reaction and as well as form structural integrity when it comes to cellular life, no? So and at this point they believe that the RNA molecule was the one who is actually the very molecule that starts the evolution of a prototype of a cell, no? So way back before eukaryotic cell, way back before prokaryotic, way back before having the cell as we know it, no, the plasma membrane covering the cytoplasm and all of those organelles, you have just a single molecule which starts the evolution of life itself, you know, and that was believed to be RNA. And this in this particular time and in this particular world, it was termed as the RNA world. You know? And the RNA world hypothesis was coined by Walter Gilbert. You know? Currently, we have three particular molecules uh, that are actually fulfilling the role of being a catalyst, being a structural molecule, and being a hereditary molecule. So we have three molecules. You have DNA, RNA, and proteins. So in your DNA, this is your hereditary molecule because this is the very molecule as to where we store our information, genetic information. Your proteins, on the other hand, they can both be catalytic in nature and structural in nature. So when you say catalytic, they can eventually hasten such chemical reactions occurring inside biological systems. No? And these types of proteins that are catalytic in nature are what we call enzymes. No? And you also have structural molecules. So these molecules helps in the architectural design of our cellular structures. For example, you have tubulin, no? You have actin, myosin, histones. No? So all of these are structural in nature. So without proteins, you can never form the cell. Diba? Your cell is being made up of certain proteins. Your proteins are the building blocks of cell. So structural in nature and catalytic in nature. When you go to RNA, your RNA are actually catalytic and also hereditary in nature. Catalytic because currently scientists have discovered certain types of RNA that can eventually splice themselves. When you say splice, meaning makat niya ang iyahang own sequence, and after cutting, it can eventually attach itself to another sequence or insert itself to another sequence or replicate itself, no, independent from the DNA. So that these particular catalytic RNA are what you call ribozymes. No? So these are your catalytic RNA. Your RNA are also hereditary in nature. Why? Because they eventually carry the information coming from the DNA after transcribing the information coming from the DNA. So, di ba, when you try to go back into your central dogma of molecular biology, you start with DNA, and then the information is transcribed in the form of mRNA, and then the information in your mRNA is translated to produce or express proteins. No, so your RNA therefore can eventually maintain itself because it can catalyze chemical reactions. It can replicate itself independent without the presence of DNA and protein, and it can also store hereditary information in its sequences. So therefore, your RNA can stand alone, no, without the presence of your DNA and protein. That is the main idea of the RNA world hypothesis, that your RNA can be independent without the presence of the DNA and protein, but still can maintain itself and can reproduce itself. No? So when you try to look at the interaction of your DNA, RNA, and protein in this particular figure, no, this is a simplified um, central dogma of molecular biology. No? So you have DNA, which would be transcribed into RNA and then translated into protein. So your protein is dependent to your RNA because you cannot produce your protein without the RNA, which carries now the information coming from your DNA. So dili ka kaya produce o protein kung walay RNA. Now, your DNA is dependent to your RNA because the information in your DNA cannot be expressed into protein without the presence of the intermediary molecule, which is your RNA, because the ribosomes cannot read the information that is being carried by your DNA. So, in a sense, if you only have DNA, you cannot eventually express it into functional proteins, meaning dili ka kakatalyze of chemical reactions. So it cannot maintain and sustain life. If you only have proteins, then what's the use of having structural proteins and catalytic proteins if they cannot reproduce themselves, they cannot replicate themselves, because they do not have the blueprint of life, no? So eventually, there is really a time, no, as the RNA world hypothesis suggests, that the molecule RNA is the starting point of life evolution, and after which the second step of having the RNA molecule is the formation of what you call liposomes. No? So your liposomes, from the term lipolipids, no? These are actually vesicles that are um, made up of kanang lipid bilayer, no? Just like your plasma membrane. So if you have plasma, if you can imagine your plasma membrane, if for example it will eventually kanang invaginate and cleaved off, forming a vesicle, this vesicle is what you call a liposome. And your liposome or can easily be created or reproduced, though you only have to have the presence of a lipid bilayer, no? And if you have RNA, and then this RNA would be enclosed in your liposome, then that is now what you call a protocell, no? Meaning uh, liposome with double lipid bilayer, and then inside you now have the genetic material. So here, you have liposome, and then you have the RNA molecule. When you have these two, you will now create the protocell, and from that protocell, since your RNA now, uh, your RNA molecule is inside, then there might be a point where your RNA is now expressing proteins. So there would be a protocell with RNA and proteins in it. And later, your RNA would now eventually produce the DNA. Now, question, kung ang RNA ang starting point, kanang intermediate siya when it comes to central dogma, why not the DNA? No, because that's the very first molecule in your central dogma that will be used to express your proteins, no? Manong dili? Because when you try to look at your DNA structure, it's more complex or more complicated compared to your RNA. Your RNA is only single-stranded, but your DNA is double-stranded. But currently, the blueprint and the storage of genetic information is mainly your DNA, because DNA gives a stable structure for your genetic information to be stored, no, compared to your RNA, which is unstable. Okay, isa raman siya ka-strand. So through evolutionary process, no, they would suggest that RNA created DNA, and that through time the DNA had become the kanang unsa ni major molecule that is intended for genetic storage, because it is more stable compared to the RNA. But again, no, we cannot really say that your RNA, or this is the factual or the right mechanism and steps in the evolution of life, because again, this happens millions, billions of years ago, and we are only hypothesizing based on indirect evidences. No? So all of these are based of indirect evidences, while direct evidence. No? Another indirect evidence would be your fossils, for instance. No? So when you are looking at your fossils, you're actually looking into prehistoric informations, no? Mga databases. Am I going too fast? Okay ra? Okay pa mo? Okay.
Okay, perfect.
Naminaw pa. Sige. Um, so, for your RNA world hypothesis, there are two main evidences, no, for this particular hypothesis. First one is when you try to look at the structure of your ATP, or your adenosine triphosphate, the monetary energy unit that we are using for us to do work, is in the form of ribonucleotide. So meaning it is very much related in terms of structure to an RNA molecule. And another evidence is that your RNA can regulate gene expression. So meaning the amount of RNA can eventually suppress or depress the expression of a particular gene. So pwede niya i-turn on or i-turn off ang gene, no? Depending on the amount of your RNA. So when we try to trace back the molecules of DNA, proteins, and ATP, no, ang ilahad yung very primordial form would always be RNA. No? So parihad yun sila og kanang murag backbone in terms of structure and as well as function. Your ATP, proteins, and DNA are eventually dependent on RNA. Now, with that being said, no, so RNA, DNA, you have genetic kanang what you call this one, genetic structures and genetic material. These genetic materials now are being used for us to create what you call the tree of life, no, or the universal phylogenetic tree. I know nga nakita ninyo during our systematic course, no. Here we can trace all of our extant and extinct species into a singular point, no, ang ato ang root, which is what you call your LUCA, or your last universal common ancestor. So this was the very organism which paved the way to the evolution of all three domains of life, no, bacteria, archaea, and eukarya. And it was believed that our LUCA is actually a unicellular prokaryotic cell. And from there, no, if you try to look at our branch here, it diversified into two major branches. One branch is your bacteria, and the other one comprised now your archaea and eukarya. So that would mean that your archaea and eukarya are quite closely related to one another rather than eukarya to your bacteria. No? So in the past, scientists believes that archaea is the most primordial form of life, no? So naguna daw ang archaea, then bacteria, and then eukarya. But it's not actually being what you call this one, being uh, supported by molecular evidences. So what is being supported is that your archaea instead of bacteria is the one that is closely related to your eukarya. When you try to go also into proteins again, no, for instance, RNA polymerase, no, the very protein that would initiate the kanang unsa ni translation process in eukarya and archaea, they do have similar RNA polymerase compared to eukarya and bacteria. So really, archaea and eukarya are quite closely related to one another. And then in this branch, no, uh, before the onset of our very first eukaryotic representative here, you have what you call the feca, or the first eukaryotic common ancestor. So your feca, no, the first eukaryotic common ancestor, is a hypothetical ancestral species which paves way to the evolution of both the extinct and the extant species of eukaryotes. And after which, at the very root of the branch of the eukarya domain here, you have what you call the LECA, or the last eukaryotic common ancestor. This is the one, no, the hypothetical ancestral representative or species that brings about the evolution of all extant species. So tanang buhi pa until now, ang ilahang pinaka last nga eukaryotic common ancestor is your LECA. But if you are going back, no, several evolutionary distance back, then you will have the first eukaryotic common ancestor, or the FECA. No? Now, how do we, no? Yeah, how do we ba? How do systematists or scientists, no, in general, create the universal phylogenetic tree or the phylogenetic tree? I know you have this kanang background na because you already know how to kanang use mega, no, or to construct your phylogenetic tree. But the most basic concept in creating the phylogenetic tree is to align the sequences coming from different organisms coming from different domains, and after aligning, determining the differences in terms of their nucleotide bases, and from these differences you infer the evolutionary distance between two species, no? And your evolutionary distance it has something to do with the relatedness of the two species and not the time of divergence. Now, for example, when we try to go here, this is a sequence of what you call an SSSUR RNA, no? Or what you call a small subunit ribosomal ribonucleotide. Wait, nawak ko. SSUR RNA is your small subunit ribosomal ribonucleic acid. Okay, that's your SSUR RNA. Taas kaayo. And your SSUR RNA is actually present in all forms of life. So, makita ni siya from bacteria, archaea, and all types of eukaryotic life forms. So, na siya sa tanan. So, in that case, nindot siyang gamiton kay pwede man nimo i-compare ang differences now in their SSUR RNA nucleotide bases so that kung pari-pariha sila nucleotide sequences, then therefore you can say that these are quite closely related, and if dili kayo pariha, then they are not related at all. No? So that's the beauty of having to use your SSUR RNA. Now, if you now have some samples of SSUR RNA from different species, when you are aligning these sequences so that you can eventually easily pinpoint the similarities and differences in their nucleotide bases. After which, if you now have differences, then that's the time you infer the evolutionary distance. So, unsa man sila ka lahi o unsa sila ka pariha, no? If, for example, the distance is very long, no, or very far from the point of origin, this is one species, this is another species, then these two species are said to be not quite evolutionary related at all. No? But if the difference or the distance is small, then that would mean that they are quite closely related to one another. And take note, your evolutionary distance is on the relatedness, no, about the relatedness of two species, never the time of divergence. No? Mura kag nagtanaw og map ba, no? Inigtanaw nimo sa map, naka sa Philippines, o you wanted to go to Manila, for example. So, there is a distance between your Cebu and Manila, no? But you cannot eventually say unsa nga oras exactly maabot ka sa Manila because you still have the traffic, you still have any other factors to consider, no? You have the exact distance, for instance, mayingon kag kuan ra siya 10 kilometers ang distance from Cebu to Manila. But the time for you to arrive exactly at Manila, kay dili dyan mo mag-point. No? Kay naama kay daghang factor. Same here. The distance is on the relatedness but never on the time kung kanusa ni diverge ang duha ka species from their common ancestor. No? So that's how you eventually create your phylogenetic tree. And because of it, no, in your phylogenetic tree, diba, you have three domains. We now have your bacteria, your archaea, and you also have your eukarya. Now, there is what you call prokaryote controversy. Sa man siya, no? So when you say prokaryote, diba, the term would eventually refer to all life forms which are unicellular, and these unicellular organisms, they do not have the presence of the membrane-bound organelle nucleus, okay, or any other organelle that are membrane-bound, no, because these are simple type of cell, no. They only have the cytoplasm, the ribosome, the nucleoid, genetic material, no any presence of the membrane-bound organelles, no vesicles, Golgi apparatus, your what you call this one, endoplasmic reticulum, wala na sila anak, no? Kay mga membrane-bound organelles, wala sila chloroplast, ang bacteria and archaea, no? But because of the advent of our technology, advancement in science and technology, as well as the creation and development of very powerful microscopes or microbiological devices, we are now able to delve deeper into the structure and architecture of your prokaryotic cells. And some scientists now had discovered that your prokaryotes, the term prokaryotes or any representative prokaryotes, they do have um, mga membrane-bound structures. No? For example, in the phylum Plactomycetes, no, ang ilang genetic material is enclosed in membrane. Although we are not calling it as a nucleus, but there is still a structure that is membrane-bound. No? So it protects the genetic material of your Plactomycetes. When you go to other bacteria, no, they have what you call anamoxosome. So these are eventually just like your lysosomes. No, your lysosomes are membrane-bound vesicles, but this time around, your anamoxosome, these are vesicles for anoxic uh, oxidation of ammonium. No, so clearly the term prokaryote cannot anymore be used to identify or collectively call all unicellular organisms with no nucleus because the boundary between prokaryotes and eukaryotes are now becoming thinner and thinner. No? Pareha na sad ni siya sa atom challenge when you go back to the time of Carolus Linnaeus wherein you only have plants and animals. So here you have prokaryotes, eukaryotes. But because of our ability to delve into the complexities more and more, no, on the complexities of our cell structure, then that boundary between being a prokaryote and being a eukaryote is now becoming very thin. And eventually, maybe in the future, wa na dyan yung boundary, then we could not anymore use the term prokaryote. No? That is why in our course, no, in this particular course, microbiology, uh, if we can, we will do away with the term prokaryote. No? What we will be using is the specific terms, bacteria and archaea. No? So, mas specify ra gihapon na ang inyong mga unicellular representatives. Now, we are done with kanang the hypothesis on asa jud naggikan nga molecule ang inyong cell, no? Most especially our unicellular organism. But then, from the prokaryote, no, bacteria and archaea, you now have the evolution of the membrane-bound organelles. And these are the very diagnostic feature for your eukaryotic organisms. Excuse me. Membrane-bound organelles that are present in your eukaryotic organisms, no, from protista, fungi, plantae, and plants, plantae and animals. Now, the very theory that tries to explain the evolution of our membrane-bound organelle is in the theory which is proposed to us by Lynn Margulis, no, your endosymbiotic theory. So when you say endosymbiotic, it is actually a symbiotic process wherein two different species are interacting with one another, wherein one symbiont is living inside the other. So that's why endo, meaning inside, symbiosis, there is an intricate relationship of two different individuals coming from different species. Now, in your endosymbiotic theory, it eventually tries to explain the origin of our mitochondria, chloroplast, as well as another membrane-bound organelle, your hydrogenosome. No? So when you have a bacterium which is non-photosynthetic and it is believed to be an aerobic respiring bacterium engulfed by a bigger host cell or predatory cell, and in time, this particular bacterium, which is not photosynthetic, had become part of the host cell, then that bacterium would eventually evolve into what you call chloroplast. So kung mag-start daw ka og non-photosynthetic aerobic respiring bacterium, no, in that particular first step, you will be ending up with the evolution of a mitochondria. But if you are starting with a photosynthetic bacterium that was engulfed by a predatory host cell, then that's the time you will be ending up with the evolution of chloroplast. Now, there is a slight modification when it comes to the starting point of a non-photosynthetic bacterium as to where uh, the modification is now the bacterium is still non-photosynthetic, but it's not aerobic respiring anymore, but it's an anaerobe. No? So meaning it does not utilize oxygen, no, for it to create ATP molecules and to create energy. So here, the modification will have a bacterium which is non-photosynthetic, and it's as well not anymore aerobic respiring, but anaerobic respiring bacterium that eventually produces energy in the form of ATP by the process of fermentation, and thus will be producing H2 or hydrogen gas and CO2 instead of producing CO2 or H2O, water. No? So in your aerobic respiration, diba, the very product would be H2O and CO2. But a slight modification, if you have the absence of oxygen, then you have now the products of CO2 and H2, no? Through the process of fermentation. So hydrogen hypothesis embedded under your endosymbiotic theory would also suggest that your hydrogenosome, which is another membrane-bound organelle in your eukaryotic cell, is a product of a symbiosis between a larger host cell and a smaller bacterium, no, which is non-photosynthetic and anaerobic in nature. Okay. There are several other, no, theories and hypotheses with regards to the origin of life. No, another one would be what you call panspermia theory. Time check, unsa na time? 3:50. Okay. So for another theory for the origin of life is what you call panspermia. I don't know if I discussed this last semester. Na-discuss ba ni nato ang mga panspermia?
Yes, sir, you've mentioned it.
Mentioning ako. So you have panspermia theory proposed to us by Anaxagoras. So here it was believed that your life form came from other planets, no, and then your asteroids are just vesicles, no, or vehicles of your life form coming from other planets going to ours. No, that's panspermia theory. Another would be, ayan, naka-remember na day ko, na-discuss din ni nato. Your primordial soup, no? Proposed to us by Alexander Oparin and John Haldane. No? So in your primordial soup, di ba, your ancient oceans, these are now referred to as the primordial soup, as to where all the ingredients for life form to be created are present, no? And with the presence of lightning, which brought about chemical reactions and energy, it now catalyzes, no, chemical reactions that would eventually combine these inorganic compounds to create organic compounds, no? So this was kanang proven by the experiment of Miller and Urey, which is the very experiment in a laboratory setting that eventually produces an organic compound. No, that's a breakthrough in science. And then you also have spontaneous generation. No? So sa inyo ang spontaneous generation, this was kanang proposed firstly, no, by Aristotle, but then several uh, years later, you have prominent scientists that are also agreeing to the idea of Aristotle that life can spontaneously be created out of non-living objects, no? And one of them would be Jean Baptiste van Helmont. So si Jean Baptiste van Helmont, he did an experiment as to where he created a formula to kanang on how to create a rat, no? So ayong formula daw kay naa kay mga bulingon, yung butangan mo grains, no, rice, corn grains, and then several days later you can create rats, no. And we all know that this is not possible because the rats there came from somewhere, onya na attract lang sila because of the presence of the food, which is in the form of the rice, corn, or the wheat, no. And then you also have John Needham. So si John Needham, another scientist which provides evidence to spontaneous generation. Ang siyang gihimo na siya flask, and then he heated the flask, no, and then after which kanang gitabunan niya og cork and flask, but since natabunan man siya, then that would mean nga walay external force nga mosulod sa flask. But then several days later, niturbid ang broth inside the flask, meaning there are certain life forms that are growing in the broth. So meaning from the broth itself, maka-create daw ka og life, no? Which is again not scientifically correct because the broth there is not sterilized. Gihit lang siya. So it does not kill all of the bacteria, most especially those bacteria that can produce endospores. No? So in that case, wala pa man sila idea with regards to that. So in that case, ang iyahang conclusion is that you can create life out of non-living materials, which is in this case the broth. No? So that's spontaneous generation. And then, uh, kanang looking into another kanang ano ni another aspect of microbes, no, really, when it comes to the reproduction of microbes, no, most especially your bacteria and archaea, we all know that your bacteria and archaea are asexually reproducing organisms. So meaning they do not have males and female individuals, they do not have gametes, and they are actually reproducing in terms of binary fission. So kalit lang sila split, motunga, and then yung nagtunga, duha na sila kabuok na two different independent and isolated bacterial cells. So therefore, when it comes to kanang mga biological concepts, most especially the concept of biological species, no, we cannot eventually call, this is not actually a right term to call our microbes, most especially bacteria and archaea, since they are really not sexually producing organisms, no? So in your biological species concept, di ba, when you say species, these are a group of interbreeding individuals that are reproductively isolated from other groups of organisms. So meaning kanang dili sila maka-interbreed with other individuals of another species. No? So that's what you call a species. However, in your archaea and bacteria, they do not really do the sexual reproduction. They maintain their genetic variation in terms of accumulation of mutations, geographic isolation, as well as what you call horizontal gene transfer, or what you call your HGT, no? Horizontal gene transfer. In your horizontal gene transfer, pwede sila mo kuha of genetic material from their environment or from another bacterial cell or from a virus. So in that case, pwede nila ma unsa ni ma differentiate, pwede mo varied ang ilahang genetic material when it comes to that. But most especially, you have the mechanism of mutation, no? Spontaneous mutation. So if there is accumulation of all these mutations, then therefore, for a group of particular bacteria, they will have a certain group of or a certain set of genotypes. No? So in biological species concept, dili pwede ma-include ang inyong bacteria and archaea. But the rest of the life forms, you can eventually call them as biological species. Now, when we go to bacteria and archaea itself, no, the more appropriate term for the bacterial and archaeal species is what you call a strain. No, so when you say bacterial species, therefore, a bacterial species is a group of different strains. Now, what are strains? Your strains are actually the descendants of a pure bacterial culture, no, with their own unique set of genotypes, own unique set of morphology, own unique sets of mutations. No? So, ang kani silang mga strains, therefore, they have similar genetic sequences, but there is a certain uniqueness in each of these groups. No? So, kung ang tanang strains nga pareha silang sequences, i-group mo siya, you can call them as bacterial species. No? So, in this case, ang pag-determine di ay sa imuhang bacterial species is not solely on the morphology, the color, the colony characteristics, but it should be with the use of genetic sequences, no comparison of genetic sequences, because you cannot eventually say that this particular bacterial colony is another species and this one is another species just because they have different colors, no. They can have different colors because they are different strains. But if you are looking now into their genetic sequences, they are very much similar with one another. Thus, if you combine them, you can create bacterial species. No? So I hope na understand na siyang apart. And when you go to strains, no, therefore, a single bacterial species, a single bacterial species can now be differentiated into different strains, and your different strains now can be described in various ways. So you can now have what you call biovars. Now, when you say biovars, these are strains that are characterized by biochemical or physiological differences. So for example, kung naka-culture nga bacteria sa imuhang agar or sa imuhang petri dish, um, na ay certain type of bacteria nga mo change ang color sa imong agar because they do fermentation process. No, naapoy some colony of bacteria mo change ang pH sa imong agar because they do aerobic respirations and so on. No, so these are kanang telltales na there is quite differences in terms of biochemical and physiological differences in your pure culture. No? So those are biovars. When you go to morphovars, so they differ morphologically. So are they coccus or bacillus or spirillus in terms of shape, no, in terms of their colony, soft ba ang iyahang edge or textured or concentric, no, and so on and so forth. So kung morphological differences ka ang makita nimo sa imuhang colony, that's what you call morphovars, no? Then you have serovars. So serovars have distinctive properties that can be detected by antibodies, no? So from the name serum, these are now your serovars. Then you have your pathovars, no? Pathogenic pathology, no? Disease causing. So your pathogenic strains distinguished by the plants in which they cause certain diseases. No? So these are just some ways on describing some strains for a particular bacterial or archaeal species. Now, when it comes to the evolution of the microbiology discipline itself, no, the very first step that paves the kanang sa ni the evolution and development of microbiology is really the availability of powerful microscopes. No, without your microscopes, dili jud ta makastudy og microorganism. No? So kung wala tong nag-develop sa inyong mga microscope, wala tatay studyhan karon. And then another thing is that your microscopy, no, and cultured based techniques, they play an important roles in terms of the evolution of microbiology because we are now able to um, kanang unsa ni isolate some bacteria from their natural habitats or natural environment and culture rare them under laboratory conditions, no. The very distinct feature in the discipline of microbiology is that your microbiologists often remove your microorganisms from their natural habitat and grow them in isolation from other microbes. That's why you have cultures that you call pure culture, no, or ascenic culture. But there is a limitation in that, no, because you are now rearing your microorganisms in isolation, meaning pure sila, walay laing bacterial species or strain in one petri dish. That would mean you cannot kanang study the entire biology of your pure culture because they are isolated from other microorganisms. So you cannot study how they behave, how they interact, the ecology with other microbes as well as their environment. No kay naaman sila sa petri dish and they are isolated. Mura siya nag kanang inyong petri dish, mura siya kanang nasa zoo ba, no? So sa inyong zoo na kay cage for one animal, another cage for another animal. But in your microbiology laboratory, one petri dish, one animal, another petri dish, one animal. So you cannot really study their whole biology, no, holistic approach, because they are isolated from one another. No? So that's one particular limitation in having pure cultures. But then again, we are now able to kanang rare certain types of bacteria under laboratory conditions, and we are able to pinpoint those bacterial species that are quite pathogenic in nature, meaning they are disease causing. Now, when it comes to the microscope, no, there are several scientists in the past that had contributed when it comes to the development and the examination of microorganisms under the microscope. No? For example, we have kinsa niya kung ibutang si Francisco Stelluti, no, si Stelluti did a microscopic observation first, no, on bees and weevils, no, in 1625 and 16 up to 1630. So he's usually working with insect specimens. Now, most especially this time, you have orders Hymenoptera and orders Coleoptera. No? And then another one is in the person of Robert Hooke, no, which eventually is a famous scientist. No? Perminin na to siyang mention sa inyong mga biology courses. And Robert Hooke was the one who coined the term cell, di ba? And he was the one who examined a cork specimen under a microscope, and he eventually discovered that the cork is made up of tiny compartments. No? So he called these compartments as cells. And he published this particular kanang particular findings, as well as his findings on examination of microorganisms in his book Micrographia. And then here comes also a contemporary of Robert Hooke. No, they are living kanang simultaneously, or they are eventually living at the same time period. No, si Anton van Leeuwenhoek, which was the one who is attributed with the development of our microscope. No? So Anton van Leeuwenhoek again is a linen merchant. He's not eventually a scientist, but because of his hobby in making lenses, so he created a simple microscope or a magnifying lens, which enables him to examine and look into the microbial world. So he was the first one who saw and examined and described bacterial or cells, no, protists, your globules or the red blood cells. He was also the very first one who examined and described a sperm cell. No? So daghan siya na discover nga micro kanang organisms as well as microbial micro structures. No? So that's Anton van Leeuwenhoek. And then, with the development of the microscopes, there is also development in certain school of thoughts. No? Diba? I've made mention about the school of thought of spontaneous generation a while ago. But because of our advanced knowledge in microbiology at this point, that there are now scientists that are making experiments contrary, no, and providing evidence contrary to the experiments that kanang agrees with the school of thought of spontaneous generation, no, such as John Needham ug si katong kinsa ni Van Helmont, no, di ba? So in this case, you have two prominent scientists that debunks, no, or provides evidences contrary to spontaneous generation. And these were in the persons of Francisco Redi, and you also have Louis Pasteur. So unsay ilahang contribute, and how did they debunk the spontaneous generation school of thought? No? So si Francisco Redi, he was the one who provided evidence against spontaneous generation in larger organisms. And si Louis Pasteur po, siya ang nag-provide of scientific evidence that there is no spontaneous generation in terms of at the level of the microorganisms. No? What happens here was that Di ba si John Needham, iyang experiment on broth, ni support sa spontaneous generation. But this was kanang unsa ni countered by the experiment of Francisco Reday as to where gibutang niya ang meat inside a beaker or inside a glass container and then put a cover or sealed that particular container with kanang cork or even a cloth, and then several days later there is no kanang creation of flies or maggots in your meat which is contained in a sealed container compared to a container nga walay tabon. So, sa katong walay tabon, then obviously nailangaw. Pero sa katong walay naitabon, walay langaw. So, that means that your meat does not spontaneously generate maggots and flies. So, that is eventually an evidence nga walay spontaneous generation in your larger organisms. No? Those organisms that can be seen with your naked eye. But here comes other scientists na ni-support sa inyong spontaneous generation nga although walay spontaneous generation when it comes to larger organisms, but there can be spontaneous generation at the microscopic level, no, microorganisms. And Louis Pasteur counteracted that particular experiment of John Needham by making an experiment using a swan neck flask. No? So, kang John Needham, di ba, iya man itong gitabunan ng isa ka container, isa walay tabon. But both there has kanang growth of life forms sa iyang broth. But in the case of Louis Pasteur's experiment, although two flasks, swan flask, they don't have covers at all, but if you break the swan neck part of the flask, then the growth of bacteria would eventually commence on the broth. No, so eventually Louis Pasteur, no, as a result and kanang major findings about his experiment, told the community, the scientific community, that maybe ang air or hangin nga nisud sa flask ni John Needham contains those kanang life forces, no, or kanang mga seeds, or in our case spores na or bacterial cells that can contaminate the unsteriled broth, no. So here iyahang gipakita nga sa iyahang swan neck flask, ang air can enter freely towards the inside of the flask. There is a continuous kanang direct contact between the broth and the air. However, the debris and the bacterial cells, the spores that are present in the air was actually filtered out at the swan neck portion of the flask. So ang made contact ra sa iyang sterile broth is only purely air. So walay spores, walay bacterial cells, kay nasangit man diri sa depression part sa swan neck part sa iyang flask. And with that, with that finding and with that simple experiment, the Louis Pasteur had completely and ultimately kanang trashed and debunked the school of thought of spontaneous generation. So, wala na jud siya ka-recover. Okay? Another thing when it comes to the past school of thought or thinking, in relation to microbes, is that they do not have yet the kanang idea that your microbes, most especially bacteria, can be pathogenic in nature. Meaning, ang bacteria makakause sila og diseases. Wala pa na sila idea ana. Kay wala pa man sila kabaw nga naay bacteria nga na-exist, no? Kay wala pa man sila microscope during that time. So they attribute certain diseases sa mga tao in the imbalance of certain body fluids sulod sa inyuhang kanang lawas, no. So this is what you call theory of humorism, no. Humor, ang kaning HUMOR, humors are actually the bodily fluids wherein the imbalances of these humors or body fluids can cause changes in a person's health, well-being, and temperament. No? So, unsa ni siyang mga humor or body fluids? You can have the blood, no, the red bile. You also have the black bile coming from the spleen. You have the yellow bile coming from the liver. And you also have the phlegm or the white fluid. So, if you have these four and you have imbalances when it comes to the increase or the decrease in terms of density of these body fluids, then that could eventually influence or affect your mood or affect your health. No? So, mo ni ilahang gi-attribute sa temperance, well-being, and health sa inyong tao. For example, if taas imong dugo, no, for mo na ilahang line ba, no, taas imong dugo, you have more red blood cells or red bile, then that is what you call a sanguine individual. No? Unsa may temperament sa inyong sanguine individual, they are optimistic daw, they are kanang positive, with positive attitude, no, mo na sila mga tao nga taas og dugo. But when you go to imbalance regarding the black bile, so the fluid coming from the spleen, then that is what you call a melancholic individual. No? So, kanang mga prone to depression, no? Sadness, deep thoughts, reflective people. So, these are melancholic. No? Kanang kabantay pod mug namang po'y tao nga murag inanak pod sila og aura, no. So taas na ilahang body fluid coming from the spleen, so active ilahang spleen organ. You also have what you call yellow bile, no? So yellow bile, kung taas, choleric ka, no, cholera. You are bad tempered, no, dali maglagot, dali sa puton, no, kanang kuhiton ra gani nagyaw-yaw na, no. So one in mga choleric people. And then lastly, you have also the white fluid. So kung daghan kag phlegm, daghan kag white fluid, it is said to be that the person is sluggish, no, apathetic, calm, no. So kanang sloth, kumbaga kanang lazy, no, inana. And they also believe that certain human diseases comes from what you call bad air or miasma, no? Kanang poisonous vapors daw. So during their time, most especially during the kanang medieval era, medieval time, the Black Plague and so on, kung naibaho nga air, they attribute the bad air miasma as kanang the cause of certain human diseases. No? So, to combat your miasma or supernatural forces, they use incense to fight off the bad air. No? So, ang original purpose jud sa atong insenso, nagigamit nato sa kanang mga ato ang misa, is actually to fight off the bad air or the supernatural forces that is present in the air that can or may cause human diseases. No? So these are what you call miasma. So in this regard, we do know that the people in the past, they really don't have the concept of bacteria as the cause of certain human diseases. No? Dili nila ma-associate or ma-connect nga ang presence sa bacteria can cause diseases sa ato ang lawas. No? So they would attribute all of these diseases to other factors, and I had mentioned and presented to you only two factors, pero daghan pa jud kay na sila, no. So two factors here, we have miasma or imbalances in the bodily fluids, no. And but since the advent and the development of our microscope, then we also are evolving, no, or kanang increasing our understanding on the biology of microorganisms, most especially those bacteria that are deemed to be pathogenic. No? So now, we now know nga dili miasma, dili bodily fluids imbalance ang nakakos of human diseases, but there are certain microorganisms that can eventually lead to these particular diseases. No? And this particular concept or school of thought is what we call a germ theory. And in your germ theory, there are three scientists that have contributed a lot, no, so that we might kanang unsa ni uplift or advance our understanding in the pathogenicity of microorganisms, most especially bacteria. And who are these scientists? You have them in the person of Louis Pasteur, no, for his kanang pasteurization process contribution. You also have Joseph Lister for his antiseptic surgery, and Robert Koch for his Koch's postulate. No, so what's your Koch's postulate? Your Koch's postulate is a very simple way of knowing kanang if a microorganism or a bacteria is the main cause of a disease, no? So mo ni siya ang gigamit sa inyong mga medical professionals to pinpoint really what particular microorganism is the cause of a disease, no? So there are four simple steps in your Koch's postulate. First one is that the microorganism must be isolated in an individual with a particular symptom of the disease, and that microorganism should be kanang isolated and cultured within a laboratory condition or under laboratory condition. Second step is that the suspected microorganism must be grown in pure culture. And then third step would be the same disease must be manifested in a healthy individual if the pure culture and new hangi culture within your laboratory is kanang introduced to that particular individual. And then the last step here is that the same bacteria or microorganism must be isolated on that healthy individual which is now diseased, no? Which is now showing the symptoms of those individuals having the same disease, and culture that bacteria within or inside or under laboratory conditions. So simple ra kay siya. Kuhaon nimo ang in general, no, ang steps in yung Koch's postulate. Kuhaon ninyo ang bacteria from an individual with a disease with a certain set of symptoms. Inikuha sa inyong bacteria, kailangan makakuha mo og pure culture ato nga bacteria. Unya introduce ninyo sa bacteria sa individual which is healthy. Ang healthy nga individual should manifest the same symptoms of those individuals contracting the disease. And then lastly, sa katong inyuhang nasakit na nga tao, inikuha ninyo sa sample the same bacteria nga inyuhang na-isolate coming from those deceased person would also be quite the same and present sa inyuhang another pure culture. So that's how you eventually identify whether a particular bacteria is the main cause of the disease. No? So that's what you call Koch's postulate. Now, aside from your germ theory, you also have what you call several scientists that had contributed to our ability to create pure culture under laboratory conditions. Now, first one, you have Fanny Hess and Richard Petrie. If I'm not mistaken, these two are married to one another, and they are students of Louis Pasteur, no, if I remember it correctly. Fanny Hess, um, she was the one who proposed to use agar instead of gelatin, no? So in making kanang solid media kanang mga agar nato, usually or previously they are using gelatin. However, with his suggestion, they converted the use of gelatin into agar, no? So they eventually got the agar coming from kanang mga algae, no? So kanang kariginan, for example, they are good source of agar, no? So why is that agar is better compared to gelatin when it comes to making or creating solid media? Your agar has a very high melting point, so about 100 degrees Celsius. So dili siya dali mo unsay bisaya sa melt, malanay, no? Nya dili pud siya dali mo gahi, no? So it will eventually require, no, about 50 degrees Celsius for it to solidify, no? So agar eventually is better compared to gelatin. Ang gelatin dali man gud siya mo gahi niya, dug kanang dali pud siya malanay, no? And so dili ka manipulate when it comes to kanang creating pure cultures of your bacteria. Now Richard Petrie from the name itself, no, he was the one who created the petri dish. Okay? Unya naapod kay another scientist, no, in the name of Charles Chamberlain. Si Charles Chamberlain was the one who eventually created what we call the porcelain filter. But this filter is unsa ni intended for the removal of bacteria. However, his porcelain filter was used by Dmitry Ivanovsky and Martinus Beijerinck in actually filtering unsa ni causative agents for the study of tobacco mosaic disease. And they eventually and accidentally discovered the presence of the virus with the use of the porcelain filter. Kay after filtering ang katuilahang plant samples, they the extracts or the plant saps itself still continues to be very infectious. So therefore, they they concluded that there is something still in the extracts and plant sap that is actually causing the disease. So it's not bacteria, it's smaller than the bacteria. No? So with the use of the porcelain filter, intended man unta siya nga i-filter ang bacteria, but accidentally they discovered the presence of the viruses. Kaya mas gagmay pa man ang viruses compared sa pores sa porcelain filter, no, na nga nang kuan pa sila, wala sila na filter out. And then you also have um, Perrault, no, another French scientist, contemporary of Louis Pasteur, which who both of them eventually developed the vaccine. So your vaccines, we already know that these are mga counter agents when you have viral diseases, no? So for example, you now have the chicken cholera vaccine, which was developed by Pasteur and Rowe. You also have the anthrax, which was developed by Pasteur and Charles Chamberlain, no, a while ago. And then you also have the rabies, no, developed by Louis Pasteur himself. So ang story sa rabies, ang first individual nga na-contract aning rabies and reportedly saved by the vaccine for rabies was actually Joseph Meister. So ang condition ni Joseph Meister kanang bata nga kuyog ni Louis Pasteur, kani nga bata, ang story mong ni Joseph was that napaakan siya giro, and then he developed the symptoms of rabies, hydrophobia, no, dili ganahan og tubig, and then ang kaninga bata kamatyonon na jud siya. So the ang weight, ang cost para i-save ang bata is more than the risk of using the unvalidated and unconfirmed rabies vaccine. Kay during this time, ang katong rabies vaccine manggod kay dili pa siya, wala pa siya clinical trials niya, murag makaingon kag kanang no therapeutic claims. But then again, si Joseph Meister kay kamatayon man siya, so unsa may mabuhat nila, no? So it's either mugamit sila sa rabies nga naay chance nga maayo ang bata or not, pasagda nilang mamatay, no? So the better option is to use the developed rabies vaccine. And after injecting the rabies to Joseph, injecting the rabies vaccine to Joseph Meister, several days later, the symptoms of the rabies subsided. So nagkaayo si Joseph Meister, and that's the time nga na-validated ang unsa ni ang effectivity sa rabies vaccine with the use of inactive forms of the rabies virus itself, no? So during that time, it's a very big news, no, when it comes to the development of the vaccine, and thus several scientists and several organizations, schools around the world had poured funds and monetary support to Louis Pasteur to create what you call the Pasteur Institute in Paris, France. So ang Pasteur Institute or the Institut Pasteur, this is the pioneering microbiology, virology, and kanang interaction of viruses and microorganism, the pioneering institute or school for the study of microbiology. No? And um, I was privileged to be at Institute Pasteur no, several years ago, and I was able to also visit the very laboratory of Louis Pasteur, no kanang naa sa picture na sa Luyo. Uh, it's a very beautiful institute, no, and every building nila naadjud sila um, Nobel Prize awardee. No, nindot, may taang si Anyu na apoy Nobel Prize awardee every building. Well, maybe naa sa inyong batch, no, wata ko balo. But yeah, so eventually the entire community, scientific community and the world had proved to be kanang unified in terms of pouring their support, monetary support to Louis Pasteur, thus establishing now the pioneering institute and school for the study of microbiology. Um, Another thing is that we also have Martinus Beering, katong ganina, no, the discovery of virus. But together with Sergei Winogradsky, they had now developed kanang different types of media. So not only the general media, which can grow almost any type or any forms of bacteria, but this time around, they developed what we call enrichment culture and selective media. So we have now mga agar nga they only kanang allow the growth of certain types of bacteria, no? So these are what we call selective or enrichment culture media. In summary, no, so the progress in our discipline of microbiology is actually the first step there is really the development of our microscope from the simple magnifying glass to a simple uniocular microscope to a compound binocular or microscope to an electron microscope. That's a very big leap, no, in terms of our advancement in the study of microbiology. Another thing is the identification of the biochemical and physiological characteristics, no, as paved way by our ability now to create pure culture of our bacterial organisms or representatives coming from the natural environment. And we are also able to compare the genetic and protein sequences of all of these bacterial species and strains so that we may now able to identify what particular bacteria is unique and what particular bacteria can be pathogenic. No? So overall, mauna siya nga to ang advancement in microbiology. And I think I will be kanang stopping here. Any questions with respect to our introductory lecture? Anyone? Wala? Or reflection? For your reflection, I'll be giving you another assignment for next week. No? So kani mo ni siya. Your assignment number 2, read and follow the instructions below. Upload your output in PDF file in the designated Google Drive folder. So in your output, write your name, the course, and the date today or the date when you started kind of creating your output. And use the file name format, family name underscore under assignment 2. So parihara sa inyo ang assignment 1 ng format. Your output will be subjected to plagiarism test upon checking. So here in this particular assignment, no, make an inventory of all the activities you can think of in your community that directly depends on microbiology and all the microbial products you often use in your everyday living. So be sure to consider all foods and medications that you are using in a daily basis. Consider how your life might be different without these items. So, unsa ang inyong kinabuhi kung wala ni sila? Write a reflection essay on how microorganisms and the discipline of microbiology affect our life as we know it. So, maximum of only 1 page. Arial 12, short size, and double spacing. So deadline for this one will be next week, Wednesday. No? Ang inyong assignment 1 and uh, lab exercise 1 would also be on kanang next week, no? Thursday. Ani siya Wednesday. Okay? Any questions? By the way, I'm waiting for the groupings. Di ba kamo man tayo groupings? Kay mag-assign na ta og reporter next week for lab exercise 1.
Hello, sir. Kanang,
Yes.
okay na di sir. Ang sa groupings po.
Sige,
Ano gyapon sir kanang same gyapon sir katong nag-voting man may about ana and same gyapon sir ang among grouping sa katong systematic.
Kanang paliw
Yes.
na lang kog send.
Yes, sir. Apo. Ang sequence sir sa grouping sir kay same gihapon. I mean,
ako'y magkuan lang ta mag fishball atong tanaw
okay.
kasi mag-una para ma-shuffle siya.
Sige sir. Mag-send ko sir sa GC sir.
Sige, thank you. Wala naman siguro, no? Yeah. So, walay concern. I'm also planning to give you a quiz next week, Wednesday. Magdalene, oh.
Ay, hello, sir.
Hello.
Am I audible, sir?
Yes.
Kanang mag-lab konta, sir. Kanang mo visit tag lab.
Face to face. Wala pa dyan ko'y balita sa una lang. Naaman daw kanang press conference tomorrow. Nakadungog mo, anak. Regarding
sa among answer kay wala pa ata.
Naando sila live press con tomorrow with the president regarding sa 100% online. So, maminaw lang siguro taugma. I don't know if i-live coverage ba na nila sa Facebook. And I also wrote a letter na kana siya, wala pa dyan balita sad. But hoping pwede ta do face-to-face na laboratory exercises kay sayang ang semester and sayang po ang skills nga dili ninyo makuha ba. Yeah, muna to siya ang update for as of now. Yes, any other concerns? Wala na. So by next week, Wednesday, kindly upload your assignment 2 sa Google Drive. Akong i-send ang link sa tong GC. And then, I'll be expecting that on Thursday, we shall have the first reporting of our laboratory exercise 1. And on Wednesday also, we will have our first quiz on Wednesday. Okay? Sige. So, I hope everyone sani well informed. Wait, sa kuon din ako ang inyuhang ay naka-share pa man day nino. Kuon ako ang inyuhang attendance. Alazas Altea, are you around? Say present if your name is called, please. Altea Alazas. Present, okay. Aplicador Brad.
Present, sir.
Okay. Elaine Arofo.
Present, sir.
Ayuda Cherry.
Present, sir.
Princess Benitez. Okay. Joy Cabajo.
Present, sir.
Okay. Maria Cadiz. Maria Victoria. Magnam to siya ganina. Wala ba? Kent Cardenio.
Present, sir.
Okay. Erica Duray,
Present, sir.
Magdalene Entise, na ato siya.
Present, sir.
AJ Galarido. AJ.
Present.
Ah,
Present. Present.
Clarence Goria.
Present, sir.
Okay. Abigail Hipolito. I think
Present,
that's
sir. Hi,
Abigail.
sir.
Okay. Hello. Zaira Limuran.
Good day, Lemoran. Present, sir.
Okay. Joanna Lugo.
Present, sir.
Present. Olive Montejo.
Present po, sir.
Okay. Zarina Negro na apo si Za.
Ay, yes, sir.
Yuko Uno.
Good afternoon, sir. Present.
Good afternoon. Christian Radin.
Present, sir.
Okay. Jonathan Ruiz.
Present, sir.
And then Terence Abilyaga.
Present, sir.
And then Onabia Nicole. Nicole, wala si Nicole. Villa Cora, Maria Teresa.
Present,
Maria
sir.
Teresa, sorry.
Present, sir.
Okay. So, ang wala si Onabia and si Cadiz. For example, si Cadiz ganina, no? Okay, anyway. So, thank you everyone for being present. Actually ay. Sige, present, Maria Victoria. Thank you everyone for being here this afternoon. So, see you next week again for our quiz and submission of assignments and reporting of the laboratory exercise. So, we are dismissed. Goodbye and thank you.
Ay, excuse me, sir.
Oo.
Kanang nasa na di ay, sir, ang member, sir, sa GC.
Ana sa messenger,
Yes, sir.
sige.
Mag-assignment na pa ba, sir, sa katong assign nga i-report
Oh,
na, sir, next
Ako
week?
lang mag-raffles ako sa inyo ang groupings anya. I'll kanang send the group number sa ato ang GC.
Noted, sir. Thank you, sir.
Thank you. Sige.
Ay, sir,
bye.
kanang, last na lang, sir, kanang, ang mag-report na next week, sir, kay ang for laboratory 1, ra. So,
On
bali-
Thursday.
Okay, sir. Thank you, sir.
Sige. Cherry, you're raising your hand.
Excuse me, sir. Kanang i-share ani mo ang atong PPT sa yung discuss today? O pagkapunta?
Ganahan mo, pwede ra. Ako pwede
Yes,
ra akong
sir.
i-share. Pero our discussion is actually kanang found sa ato ang reference book. That's chapter 1. So for any additional information, you may also read chapter 1 of katong Wiley et al. 2020. Okay? Sige.
Okay, thank you, sir.
Hi, sir.
Okay,
Last
bye-bye. Ay,
clarification lang, sir. Mag-quiz. Mag-quiz next week, sir, no?
Wednesday.
Okay. About ani...
About kanilang chapter 1.
Okay, sir. Thank you.
Okay, bye-bye.
Bye, sir. Thank you.
Bye, sir.
Thank you, sir. Goodbye.
And thank you so much, sir.
Thank you, sir.
Thank you, sir.
Bye-bye.
Thank you, sir.